Hello student, good morning. I am Dr. Ajay Kumar Sinha, Professor of Chemistry. Today I am going to discuss function of insulin in our body. Function of insulin in our body. What is insulin? That is the first question. Then insulin is a hormones. Then now next question is what is a hormones? Then hormones are chemical substance that act like messenger molecule in the body. After being made in one part of the body, they travel to the other part of the body, where they help control how cell and organ do their work. For example, insulin is a hormone that's made by the beta cell in the pancreas. Means insulin is hormones. Correct? And what is the hormones? I have told you. Again I am repeating. Hormones are chemical substances that act like messenger molecule in the body. After being made in one part of the body, they travel to the other part of the body, where they help control how cell and organs do their work. For example, insulin is a hormone that's made by the beta cell in the pancreas. Now I will go little details about insulin. Insulin was first isolated in 1922 from the pancreas of dogs by two scientists, Banting and Best, both of the University of Toronto, Canada. The chemical structure. Here one more things you can understand. These two scientists also demonstrated the curative effect of pancreatic extract in dogs ailing with diabetes mellitus. Means dog also becomes diabetic. Okay. Then these two scientists, Banting and Best, in 1922 demonstrated the curative effect of pancreatic extract in dogs ailing with diabetes mellitus. The chemical structure, or you can tell insulin is in fact the first hormone to be recognized as a protein. This is a very, very important point. The insulin is in fact the first hormone to be recognized as a protein. The chemical structure of insulin has been determined by two scientists. Sanger, main scientist was Sanger. The chemical structure of insulin has been determined by Sanger and his co-workers at Cambridge, England, in a painstaking 10 years from 1944 to 1954. It took 10 years time to study. Pancreas has a molecular formula C254, H377, O75, N65, S16. See, it is a very high molecular mass. Insulin is a very, very high molecular mass substance, hormones. Okay, it has a molecular formula C254, means 254 carbon atoms are there, and hydrogen is in 377 number, 377 hydrogen, and oxygen contains 75 atoms, nitrogen 65 atoms, and sulfur 6 atoms. Hence its molecular formula is written as C254 H377 O75 N65 and S6. Means there are the 5 elements which are found in insulin: carbon, hydrogen, oxygen, nitrogen, sulfur. Carbon is in 254 number, H is 377, oxygen is 75, nitrogen is 65, sulfur is 6, and all are combined to each other. I will tell you little details, but now let me tell related to insulin. Okay, just to recall bovine insulin. Now you might have heard the name of insulin also. Nowadays this word is insulin is common all over the world. Then just to consider bovine insulin, it consists of 51 amino acids. This is a very, very important point. Insulin consists of 51 amino acid residue dispersed in 2 chain. Structure, now I am telling you that insulin that is dispersed in 2 chain. 2 chains are there, chain A and chain B. Okay, 2 chains are there. Then these 2 chains are polypeptide. Polypeptide. The 2 polypeptide chains are held together by cross linkage of 2 disulfide bonds. The acidic chain A, capital A you can tell, the acidic chain A contains 21 residues, and the peptide chain B, capital B, have 30 residues, means 21 plus 30, 51. In insulin there are 2 chains. They are peptide chains. The 2 peptide chains are held together by cross linkage of 2 sulfide bonds, means 2 sulfur atoms. Through 2 sulfur atoms, these 2 chains are linking, making a link with these 2 sulfur atoms. Correct? Then the two polypeptide chains are held together by a cross linkage of 2 disulfide bond. The acidic chain A contains 21 residue, and the peptide chain B having 30 residues. Here actually I want to tell about the amino acids, but let me tell little more, then I will come to some you can say common terms. Okay, then I told about that there are 2 chains. Acidic chain A contains 21 residues, and the peptide chain B having 30 residues. Insulin has a molecular weight of 5733, very high, very high. Insulin has a molecular weight 5733 and is isoelectric at pH 5.4. Human insulin, however, has a molecular weight little less, 5800 808. Insulin is destroyed by alkali. You remember, very very important point. Insulin is destroyed by alkali, but is relatively stable in acid solution. Reduction of disulfide bond. Reduction of disulfide bond results in a loss of biologic activity. Actually, insulin has zinc also, zinc, Zn. Zinc is always found with this hormone, but is not a part of the insulin molecule. It's a very very important point. Zinc is always found with this hormone, but is not a part of the insulin molecule. And further I am telling you, insulin is a protein hormone crucial for maintaining blood sugar at appropriate levels. Chains of amino acids in a specific sequence, means primary structures, define a protein like insulin. These chains fold into well-defined structure, means tertiary structure is well-defined structure. Okay, these chains fold into well-defined structure, means tertiary structure. In this case, a single insulin molecule, this only in one insulin molecules they have tertiary structure. Okay, these two chain I told you, chain A and B, these chains fold into well defined structure, that is the tertiary structure. In this case a single insulin molecules is formed, or in a single molecules this tertiary structure is found. Such structures assemble with other chains to form arrays such as the complex of 6 insulin molecules. Correct? Here I told you only in one long very this already told you molecular mass and formula, very high molecular mass. Then molecule is very complicated, very complex, and it has a tertiary structure, 3-dimensional structure, but it may be in 4 also, quaternary. Now actually before discussing more, I want to tell you about the some basic terms like peptides. Then what is actually peptide? Peptide hormones already I told you, but peptide, just I want to tell you what is actually peptide. It is peptide hormones also. Insulin is also called peptide hormones. Then here actually peptide means peptin. This word came from the word peptin, means to soften or to digest. 2 or more amino acids, now listen carefully, 2 or more amino acids covalently joined by peptide bond. 2 or more amino acids covalently joined by peptide bonds. Then now we'll ask, what is peptide bond? Then now we listen, peptide bond, a substituted amide linkage between the alpha amino group of one amino acid and the alpha carboxylic group of another, with the elimination of a molecule of water. Then when these 2 amino acids are linking, here I want to tell you about the amino acid also. Actually what is the amino acid? Amino acid has a major role in our body also, or in insulin also. Actually everywhere amino acids are there. If you will talk about the protein, amino acid is there. Here linkage peptide also, amino acid is there. Hormones also, amino acids are there. Correct? Everywhere amino acids are there. Okay. Then here you should know what is actually amino acid. Okay. Amino acid structure you can understand very easily, that with carbon has tetravalency, you know. Carbon has tetravalency. Then one valency is used, out of 4, 2 is used up to make a bond with carboxylic group COOH, acid group. One is attached with acid group, that is the COOH. Second is attached with NH2, and third one is attached to alkyl group R, and fourth one is attached to H. Means it is just like a chiral carbon, you can tell. All different groups are there. With carbon, four groups are attached. All are different. One is carboxyl group, other is CH2 group. Correct? And third one is H. Fourth one is alkyl group. Here I want to tell you that generally people will know that there are only 20 amino acids. Generally you will ask from anybody who knows chemistry, they will tell 20 amino acids are there. But fact is different. There are many amino acids, but actually 20s are synthesized. It is there, but only 20s are synthesized by scientists, chemists. But nowadays 22 are there, 2 more synthesized, 22. Now it is 22. But 20 amino acids are taking part in coding. 20 are sufficient. But in coding, 63 amino acid can be used. 63. Then what are the 2 new I am telling you? That is the selenocysteine. Selenocysteine. SELENO. Seleno, CYSTEINE. Selenocysteine. SEC, its abbreviation is SEC, and its molecular weight is 168.053. Okay, and other is pyrrolosine. Pyrrolosine, from pyrrol, PYRROLSINE, pyrrolosine. Its abbreviation is PYL, and its molecular weight is 255.31, and these 2 are found in proteins. Okay, proteins. Actually I told you 20 are specified by the universal genetic code, and this I can say up to 63 amino acid can be coded. Okay. Now this maximum, if you want to know how many amino acids are there, maximum, then it is approximately 500 amino acids. 500 amino acid approximately. Then you will ask how. Then now you understand how you are getting so many amino acids. Then for that you have to see the amino acid structure. I told you now that carbon is attached with four different group. One group is COOH, carboxylic group, second is NH2, and third is alkyl group R, and fourth one is H. Then now you know that now alkyl group may be of only one carbon, methyl. It may be of two carbon, then ethyl. If three carbon, propyl. If four carbon, butyl. Five carbon, pentyl, means different methyl group may be there. Not only that, whenever new structure will be there by increasing one carbon, then that amino acid will be different from others. But one thing always you remember, if you are talking about amino acid, then always whatever type will be there, carboxylic acid and NH2 is common. Every time in all amino acid you will find COOH group and NH2 group. That is common. Then what will change? Then this group, alkyl group, in that how many carbons are there? If for one carbon change, if one carbon is there, then that will be different amino acid. If two carbon is there, then that is a different from first one. If three are there, then again that will be different. Four is there, again different. Five is there, again different. Every time if the number will increase, that will be called different amino acids. Not only that, sometimes cyclic, that in alkane you know that H bonds are there. That if H bond is again as a branch, it can join with other alkyl group. Branch chain it can join. Then again it will be another type of amino acid. Sometimes it can join with hexagonal benzene like structure. Then that time different type of amino acid will be there. Sometimes 5-membered ring it can attach to the 5-membered ring. Then again another type of amino acid. Like that way, that due to this you can say differences, many amino acids are possible. Then 20 amino acids actually are common, and their name is actually abbreviated. Then what are the 20 amino acids? I will tell you. Actually it is divided into two groups. Amino acid is very important in our body. It is the building material for protein, you know. Building material for protein. Protein is formed from that. I know the role of protein in our body. Hence, if we are talking about the body, then first you should understand about the amino acids. Then there are 20 common that I am telling you. Already 2 I told you, 21 and 22. That is a new one. But 20, in that also division is there. One is essential, 10 are essential and 10 are non-essential. Okay. Then what are the names I am telling you? First essential I am telling you. Essential like valine. Second is isoleucine. Third is tryptophan. Fourth is methionine. Fifth is arginine. Sixth is leucine. 7 is phenylalanine. And 8 is threonine. 9 is lysine. 10 is histidine. These are the 10 essential. Essential means here actually many amino acid is formed in our body. Because it is formed in our body, you have to not bother. Then you have to bother for, you can say, other 10 which is not produced in our body. That is actually called essential. You have to take from the outside. Then this 10 name I told you, that is essential. Now non-essential means which is produced in our body. Then that their name I am telling you 10, that is the glycine, trypsin, proline, cysteine, aspartic acid, alanine, serine, hydroxypropine, cysteine, glutamic acid. These 10 again I am telling you non-essential. Glycine first, trypsin second, proline third, fourth cysteine, fifth aspartic acid, 6 alanine, 7 serine, 8 hydroxypropine, 9 cysteine, and 10 glutamic acid. Then this I told you amino acids, and due to these amino acids, now different you can say their combination. Actually all this is the I told the name, but abbreviation is there in shortcut. Okay, just like I told you selenocysteine, SEC, correct. Also I told you pyrrolysine, PYL. Then these are the abbreviation. Like that way all amino acids are abbreviated. Then these amino acids can join to each other and can make a big molecules, complex, correct. And due to their when they will join, then how they will join? Then for that purpose I am telling you, suppose for your understanding that you consider 2 amino acid. Then in amino acid it is just like a deuter ion means positive and negative ion both are present. Then now first you consider one amino acid. Then amino acid has COOH group. If H will go, then COO minus will be there, negatively charged. Okay, then this amino COOH group, first molecule, and second NH2 is there. Second amino acid will take then NH2 end, and first one COOH. In both group, in both amino acid, both group are there, but here role is coming for one first amino acid COOH, second amino acid NH2. Then this COOH and NH2 will come close, then it will form H2O, COOH, that H, and here NH2, then OH, COOH is there, then from that OH and H from NH2, it will form water molecules, OH plus H, okay. This is H is from NH2, 2 NH2 is there, NH2, then one H from NH2 and OH from COOH, it will form water, OH plus H, OH minus H plus, it will form water, and O will be here. When OH will go, then only one oxygen will remain, COOH, and C and double O, that is a double bond, because OH went by taking its electron, and carbon will be positive. Carbon will be positive because OH went as a negative, then carbon will be positive, and this side NH2 H, then here H went as a H plus, then N will have positive charge, then negative carbon and positive negative carbon and positive nitrogen both will join, then they will make, both will make one bond, they will link, now they are making link, bond. That bond is called peptide bond, molecule is called peptide, and bond is called peptide bond. Like that way many amino acid can join to each other. But here also, because many combinations are possible, different type of amino acids are there, then they can combine. Actually bond will be every time, this type of bond will be there, but because in different amino acids are there, alkyl groups are different, hence they will make different type of protein. This structure, long structure of amino acid joined to each other, then long structure, that will be molecular structure, that is actually protein. Then now I can say what type of protein is that depends upon what type of amino acids are joining to each other, correct? Then now I have told you about the amino acids and peptide. Main things I have told you, and this role of amino acid you know well, okay? Then now I want to tell about these things, how this insulin functions. How does insulin function? Okay, then that I am telling you now, that insulin molecular structure already told you. Molecular formula of insulin already I told you. C254, H377, O75, nitrogen 65, S6, means 5 atoms, 5 different atoms, carbon, hydrogen, oxygen, nitrogen and sulfur, forms the insulin. Then already this I told you about the molecular weight. Molecular weight is 5733, correct? Insulin has molecular formula 5733, very high, and is isoelectric at pH 5.4. But human insulin has a molecular weight little less, 5808. And also I told you insulin is destroyed by alkali, but is relatively stable in acid solution. Reduction of disulfide bonds result in loss of biologic activity, correct? Also I discussed zinc is always found with these hormones, but is not a part of insulin molecules. Okay, now further I want to tell you that how will you get protein, not protein, insulin, how will you get? Then you will get this insulin from the proinsulin. From the proinsulin you will get insulin. Then what is actually this proinsulin? Then proinsulin actually is the starting material to get the insulin. Okay, it is also called precursor. Proinsulin is called precursor, and this precursor this proinsulin, it has 84 amino acid. You remember, you will get insulin from proinsulin, bigger unit. It has 84 amino acid, but insulin has only 51 amino acids. 51, you remember. Insulin has 51 amino acids, but proinsulin, which is a precursor of insulin, you will get insulin only from proinsulin. It has 84 amino acids. Then this proinsulin in the presence of proteolytic enzyme, in presence of proteolytic enzyme, there will be cleavage of bond, cleavage of 33 amino acid connecting peptide chain. This bond will break. This 84 I told you amino acids are there in this thing, proinsulin. Then 33 amino acid containing peptide chain will be separated, will break, cleavage will be there. Okay, then cleavage of a 33 amino acid connecting peptide chain from the proinsulin molecules leaving behind insulin. Means now it was 84, then out of 84 minus 33 you do, 33 gone, then how much is remaining? 51. 84 minus 33, 84 minus 33, 51, that is the insulin. Means proinsulin in presence of proteolytic enzyme, it will form insulin plus peptide. Insulin plus peptide. Now understanding the peptide also, I told you. Then this is the formation of insulin. Okay. Now what is actually these things? Its function that I am telling you now. Okay. Insulin has a profound influence on carbohydrate metabolism. It facilitates entry of glucose and other sugars into the cells by increasing penetration of cell membranes and augmenting means it will make a bigger in size will increase. Okay, then here you have to understand more that insulin is a profound influence, already told you, on carbohydrate metabolism. Metabolism means a set of reaction to get some important product. Okay, then insulin has a profound influence on carbohydrate metabolism. Metabolism is a set of reactions so that you will get desired product. Then it facilitates entry of glucose and other sugars into the cells by increasing penetration of cell membranes and augmenting phosphorylation of glucose. It will form phosphorylation of glucose, means it will form, that is the molecules, means it will due to the glucose their phosphorus size will increase, that is called phosphorylation of glucose. This results in, if glucose will come, how actually from the in blood sugar, sugar is there, we want to remove the sugar from the blood, then what will happen? This happens due to this step only, phosphorylation of glucose. Glucose will come from the blood and it will attach to the phosphorus, that is called phosphorylation of glucose. Their size will increase and in blood sugar will be less. This results in lowering the sugar content of the blood. This is the very important step. I have told you how I can say that insulin, how it is working, that main step I am telling you, that glucose, this insulin, you are using insulin, then this reaction actually takes place, phosphorylation of glucose. This results in lowering the sugar content of the blood, a fact leading to its common name. Actually it is also called Hg factor. Hg factor, small h, capital G, Hg factor, that is the hypoglycemic factor. Hypoglycemic factor. Then here this important step I told you that how it is controlling the sugar or glucose present in excess glucose, how it is becoming less. That step I told you that it facilitates entry of glucose and other sugars into the cell by increasing penetration of cell membrane and augmenting phosphorylation of glucose. Insulin administration promotes protein synthesis. Insulin administration promotes protein synthesis by assisting incorporation of amino acids into protein. This effect is not dependent on glucose utilization. At the same time, it also acts as an anti-proteolytic agent. Okay, means discourage excessive breakdown of tissue protein. This action is similar to that of GH and testosterone. This is separate subject, I don't want to tell here only just to know that insulin administration promotes protein synthesis, that is also called proteogenesis by assisting incorporation of amino acids into proteins. This effect is not dependent on glucose utilization. Okay, and also you should know that this insulin, more about the insulin, that in general way I am talking about, the insulin promotes anabolic processes. Anabolic, what do you mean by anabolic process? Means synthesis of glycogen, fatty acids and proteins. Okay, insulin promotes anabolic process, encourage this process, means synthesis of glycogen, second fatty acid, and third protein, and inhibits means stops or becomes slow down, and it inhibits catabolic ones. What is the catabolic process? Then breakdown of glycogen and fat, means it becomes slow or stops also. Okay, it inhibits means insulin inhibits catabolic ones, means breakdown of glycogen and fat. Now here I want to tell you that in pancreas, actually insulin secretes from pancreas. Correct? In pancreas, if you will see the structure I have discussed, that in that there will be two type of cell, alpha cell and beta cell. Two types of cells will be there. Okay, then these two type of cells plays a big role. Okay, these two type of cells plays a big role. Alpha cell and beta cell, what is their role? I am telling now. While the beta cell produces insulin, the only blood glucose lowering hormones of the body. This beta cell role is that it secretes or it forms, while the beta cell produces insulin, the only blood glucose lowering hormones. It decreases the glucose. Okay, it produces insulin. This is the only blood glucose lowering hormones of the body. Then what alpha cell will do? The alpha cell releases glucagon, 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 which elevate what it will do. Role of alpha cell, which alpha cell actually elevates blood glucose. Means this alpha cell will, if in blood suppose glucose is becoming down, less, then that time alpha cell role will come. It will send the glucose to the blood. Then alpha cell, its main role is just it will send the glucose in the blood. If in blood glucose is becoming less, that is also diabetic, sign of diabetic, correct? Glucose is becoming less. Other sign, that more glucose. Then alpha cell will send the glucose. Okay, the alpha cell release glucagon, which elevates blood glucose, and it's a molecular weight of glucagon, I am telling you, that is C24H42O21. This is also heavy, carbon 24 atoms in one molecule of glucagon. 24 carbons are there, 42 hydrogens are there, 21 oxygens are there. Okay. The alpha cell secrete glucagon as a response to low blood glucose. The major function of glucagon is to release glucose from glucagon stores in liver. In liver, actually, glycogen you will get this glycogen. Actually, this you will get glucose. You will get from glycogen. It is stored in liver. Correct. And this is glycogen, that is called peptic hormone, and this is called peptic hormone. Okay. Then this I have told you about these things, alpha and beta cell and their role. Glucagon is a 29 amino acid polypeptide. More about the glucagon. Is a 29 amino acid polypeptide. The polypeptide has a molecular mass, it is very high molecular mass, 3485 Daltons. Its unit is D, Daltons. 3485, very high. Okay. Now general, I know, general you might be knowing also that these things, insulin deficiency, means I want to tell you about the insulin deficiency. What will happen if insulin is less in your, means beta cell is not working properly and insulin is less, then what will happen? That is called insulin deficiency. The deficiency of insulin caused either by inadequate insulin production or by accelerated insulin destruction lead to diabetes mellitus in man. Okay. This disease is characterized by some symptoms are there. How will you understand that you are diabetic? Then first is that an increase in blood sugar or glucose, that is also called hyperglycemia, from a normal value 80 mg per 100 ml of plasma. Minimum it should be 80 mg per 100 ml of plasma, means blood. In blood, plasma will be there. And it should be maximum up to abnormal value ranging from 150 to 200 mg per 100 ml of plasma. Means in blood, glucose should be minimum 80 mg and maximum between 150 to 200 mg per 100 ml of plasma. Second, these are characteristics I am telling you. Second characteristic, the appearance of sugar in the urine. that is also called glycosuria, with the result of victim's urine test sweet. Urine will become, the test will become sweet. It means sugar is present in urine. Third characteristic, an increase in concentration of ketone bodies in blood. That is also called ketonemia. In blood, ketone will increase. An increase in concentration of ketone bodies in the blood and in the urine. If it in urine it is more, ketone is more, then that is called this ketone urea. Okay. Now fourth characteristics I am telling you, that is the excretion of large quantity of urine. You will relieve more urine. That is called excretion. The excretion of large quantities of urine. That is also called polyuria. Frequently at night. That is called nocturia, leading to dehydration. In your body, water will be less. Now next important characteristic, the excessive drinking of water. You will drink more water. That is called polydipsia. On account of an unrelenting thirst. Because you will feel thirsty, I will drink more water. Because again you are releasing more urine, you will require more water. Now next point is that the excessive eating, you will also feel more food. You will require more food. Okay, hungry. The excessive eating due to feeling constant hunger. This is because the tissues cannot utilize glucose normally, even though they need fuel. Okay, this excessive eating due to feeling constant hunger. This is because the tissues cannot utilize glucose normally, even though they need fuel, means food. And last point I am telling you, the lack of energy, that is also called asthenia. The lack of energy which is apparently caused mainly by loss of body protein. In body who is diabetic, there will be loss of protein. Then if protein is less, you will not get the energy. You know that protein provides the energy. The lack of energy which is apparently caused mainly by loss of body protein. Now I am stopping only here. Thank you.